Welcome, everyone. Before we begin, I would like to say a huge thank you to Litmaps, which is our first official sponsor of the podcast. Litmaps is a wonderful company I've been blessed to collaborate with for the last couple of months. And what Litmaps essentially allows you to do is to choose your selected papers, whatever research field you are in, paste it into Litmaps, and get a beautiful, wonderful map, a visualization of how those papers are connecting to the rest of the field. So one particular use case I found to be super useful at the beginning of my PhD when I was just entering this field of neuroimmunology was to get a broad overview of the field. So what I did was I found the biggest review I could find that was written in 2007 by the leaders of the field. I pasted it into Litmaps, and it gave me this wonderful map of all of the papers that are heavily citing my review, but that have come out since the last 15 years. So I got my beautiful big review plus all of the updates. Then I click monitor, and now I get updates on all of the papers that come out but are super relevant to what I'm looking for. So check out Litmaps. They're awesome. Their new user interface is also very nice. Super easy to use. There's a free option. If you want to upgrade to the paid option, you can do that. But by all means, you're not obligated to do so, and you can still get tons for free from Litmaps. So the link is in the description. Check them out, and let's get on with the podcast. All right, everyone. So today, in the inaugural episode of the Better Biologist podcast, I am speaking with Kajs Hadzic. He's one of my favorite people on LinkedIn. He's a research-oriented master's student, and he's mostly interested in nutrition, especially molecular nutrition and the immunology. And especially here, there's a part where he says he's interested in IBD. What is IBD, guys?
Okay, first of all, thank you for having me and thank you for the very flattering introduction. And yeah, IBD is an abbreviation for inflammatory bowel disease, basically covering ulcerative colitis on one hand and Crohn's disease on the other. There might be some others that we're kind of speculating, like certain types that we can't necessarily diagnose, indefinite IBD, where we don't necessarily put them into the colitis category, nor do we put them into the Crohn's category, but it's kind of a subcategory on its own because we don't have enough data for now. But yeah, as to me, as you already said, studying nutrition, nutritional scientist by training, mainly focusing on molecular nutrition and also going into the immunology side of things. Or one could say I'm attempting to break into like the immunology side of things because not yet there, but hopefully that will change with my PhD that will hopefully commence soon or within this year.
Awesome. All right. So the reason I wanted to talk to you is mostly because you're a pretty chill guy and you share a lot of good advice for students on LinkedIn. And you already got very far in your research career, even if you're just, still a master's student. So you're still not doing your PhD, but you already had your first paper published recently as the first author. Congratulations on that. And it's exactly about everything we mentioned right now. So maybe if you can tell me, how did you get interested in this, the IBD, the molecular side of nutrition and everything you're studying?
Well, it really depends on where we want to start and how far back we want to look at. Like, I can give you a story. Since the day I was born, I wanted to be a scientist. That won't be completely true, but almost. But the funny thing about me is, like, my entire family is, like, academically oriented, which is obviously a huge benefit for anyone growing up. All of them are either within academia or within some kind of an educational system, be it high school, elementary school, and there's a lot of teachers in my family in general. And as I grew up, it was always interesting to look at the world through a very critical lens. I guess Richard Feynman-esque would be a way to describe it, where I looked at something, I wanted to know more about it, I asked questions, and later on, I decided to do some digging on my own. So that aspect of what critical thinking and curiosity was always there, and it developed over time. But at some point during high school, so at the time when I was like 12, 13, 14, or no, sorry, 16, 17.
That's an early high schooler.
Yeah, didn't skip elementary, don't worry. Wasn't that smart. But at 16, 17, there were like two choices. Either I wanted to go into dentistry, which is kind of very radical to what I ended up doing, or I wanted to go into science. And the decision was made, I want to say at like 17 or even at 18, because I wanted to go into science at that point, but I'm not sure, or I wasn't sure if I wanted to go into like sports science because I have an extensive sports background, like 14, 15 years of high-ish level competitive swimming, or if I wanted to go into the other aspect, which was obviously very important for sports, which was nutrition. And the one thing that decided really on my future, and I waited as best as I could at that point, was I knew that I was never going to be able to get into a lab outside of academia in the capacity I needed to actually get the knowledge, get the know-how, get the equipment I need, because we're obviously aware that we're like very blessed to have millions of dollars, euros, whatever currency you'd like to use, of equipment at our disposal at like top level academic institutions. And growing up through sports, I also knew a lot of people who went into sports, and they were somewhat disappointed, but also somewhat relying on non-academic, non-traditional ways of education, like certifications and courses and so on and so forth. So if I ever wanted to go the sports route, I could do it after I did like my traditional academic route. And at that point, it was like, okay, I'm going into nutritional science. I went into it. I went to Vienna, started my bachelor's, finished it after a very long time. We might touch on that a bit later. Got into the master's, and I got the opportunity to work on a major project at our lab, which kind of started when I joined for my master thesis, which was our topic focusing on restrictive diets. And in my case, it was the impact of restrictive diets on, well, in our case, ulcerative colitis. More specifically, it was a mouse model. It was a chemically induced colitis where we induced colitis through DSS, dextrane sodium sulfate, and then we fed them specific diets to see if there is an impact, because we didn't know. Like, we had some speculation based on some previous experiments, because obviously it didn't go in fully blind. That would have been a waste of time and resources, but we weren't sure if those effects would kind of outweigh the potential downsides of a restrictive diet in that setting.
Sorry to interrupt there. Just when you say restrictive diet, does that mean restrictive in terms of calories or a particular nutrient, or do you kind of encompass all of that in a larger screen, let's say on mice?
In our case, it was really like the whole definition of a restrictive diet. So it's either a restriction in energy intake, which is like the traditional caloric restriction when you want to lose weight. It's either a temporary alteration in the eating schedule, which would be something like intermittent fasting, or it could also be a restriction of certain nutrients. The most popular example in that case would be like a keto diet where you omit carbs. And we tested a caloric restriction, two types of a keto diet, intermittent fasting, and something called a fasting mimicking diet, which is not novel at this point, but still probably quite unknown. Walter Longo, a Italian-American scientist, I believe he sits in California currently, is the inventor of that diet. And as the name suggests, you should kind of mimic the effects of fasting without actually fasting. So it's like a super specific combination of certain nutrients and certain foods and everything else. And in our case, we got it delivered from him because, or at least we got like the specifications from him, and we had to mix it and all sorts of stuff to make it like very specific to get the proposed effects of fasting. And at the end of the day, like, to summarize the broad spectrum of results, all diets had an impact, which is, well, surprising to say the least. All diets showed anti-inflammatory properties, because one of the main issues with colitis is like inflammation locally in the gut, or, well, specifically in the colon, as well as systemic inflammation that kind of spreads across the body and like ruins everything. So from caloric restriction up until the keto diet, we had similar effects, but the keto diet seemed to work a bit better than others. which will also be published hopefully within the next couple of months. We'll kind of finish the second manuscript on that as well. So I'm kind of spoiling some stuff, but yeah, hopefully people get some insights, which wasn't necessarily surprising. But the one thing that was surprising is all of the other diets, like caloric restriction, was obviously restricted in energy intake. Intermittent fasting was also indirectly restricted in energy intake because they're only eating in a certain window and they don't have like their regular eating schedule, so they don't eat as much. And keto was still fed ad libitum, so they weren't restricted in any way. So maybe even that aspect of them being able to consume the food at all times had an impact, but that would have to be obviously kind of analyzed. We didn't do post hoc analysis just yet, but it would be interesting actually to note that and to expand on that a bit further.
Sorry, so when you restrict diet from a mouse, let's say you give them a time frame when they can eat. I don't know, is that programmed in the mouse cage, or there's a person that runs around giving food at specific times? But is there in the mouse, is there an element of learning that there's a window and then going into overdrive with eating? Like, I'm going to eat 2 hours. I have 2 hours to eat. I have to eat as much. Does that curve exist in mice?
Don't quote me on this one, but I do believe it does from our anecdotal observations as well as from some data that I've seen. So basically, prior to this, we did a lot of restrictive experiments because, as I said, restrictive diets were kind of the focus of the lab. So anytime we had like a caloric restriction, there would be a person, the animal caretaker or someone from our lab or someone from the other lab if we weren't able to go in, who would feed every single mouse like a specific amount of food. And our restrictions in the case of caloric restrictions were about 70 to 80% of your normal intake, which adds up to like just as a fun fact, I guess, 2.6 to 2.8 grams of food for black 6 mice, which is not a lot, but like considering their body weight is like 20-ish grams, that's like 10% of their body weight, which is still quite a bit given their total body weight. But then again, we go into some other stuff like the proportion of their energy intake to their metabolic rate and the proportion of metabolic rate to animal size, blah, blah, blah. Won't bore you with that for now. But the thing is, we observed that at certain periods, we also had refeed periods with the mice in other experiments. And you would definitely see them chow down on the food like there's no tomorrow. So I would assume that there is some learning period because they're definitely very smart animals. Like just observing them in their cages, might seem barbaric at points, and we can get into that whole discussion of animal welfare and the need and use of in vivo experiments. But I would assume that there is some aspect where they can kind of figure out, okay, I'm not going to be fed, so I'm even going to keep it. And even when you feed them like those 2.5 pallets of food, well, if you do this 2.6 grams of food, they will split it up for themselves. Like they won't chow down immediately. Sure, they'll grab like one piece, eat it, and then the other piece will either remain in the feeder or will be stashed away in their cage. So some sort of psychological temporal adaptation is certainly there. And if you consider that with their like pronounced circadian rhythm, just like with humans, then there's definitely going to be an effect.
So mice are nocturnal. Do you try to control their circadian rhythm by reversing keeping them in a room where it's adjusted to human circadian rhythm so that you don't have to go at night?
Yeah. In that case, we're again kind of making it easier for ourselves, which again could be discussed as a potential co-founder. I believe most mice experiments in that case are done exactly that way. You keep them on a 12-12 cycle. So 12 days, 12 hours of day and 12 hours of night, which are basically exactly reverse to their usual rhythm. But given that the rooms basically have no windows or anything, or the windows are taped off, they are regulated by the artificial light within the room. So our rhythm was like 7 in the morning till 7 in the evening was their light cycle, and any experiments that should have been, could have been, would have been done, feeding, cleaning, interventions, gavaging, whatever you should have done was done that period, and then they had like their nocturnal period during our nocturnal period, because I don't think anyone was ready to go in at like 2 a.m. to accommodate for their rhythm, which would have been possible, I guess, but we struggled as it was with this rhythm, at least in our case, because we were a small lab. And if someone called me up at 3 a.m. and said, "Hey, you got to feed the mice," probably no.
I would hope that the mouse community to have some kind of programmed cage, I guess, at least for food delivery, to be like, okay, just dispose this much food at 3 a.m. so I don't have to come. Is that used in bigger labs, or is it always people just coming to feed the mice manually?
The labs I worked in weren't like huge, so our mouse house was like, I want to say around 300-ish animals, which sounds like a lot, but then you have group cages where you have like 5 mice per cage, so you have like 50 cages, and that's already 250 animals. Maybe we had more, but let's say 500-ish. And at the other lab in the Medical University of Vienna, where I work in the Center for Cancer Research, I never actually went to the mouse house. I only like got the dry training for some reason. We were split up in a weird way where I did just in vitro stuff. But that facility was a bit bigger, but in that case, it was also done by hand. So as far as I'm aware, those two labs did everything by hand. I'm not aware if, and if so, which labs in Vienna might do it automated, if any. That would be kind of outside of my current scope of knowledge. But now that you mentioned it, I'm definitely going to figure it out just for myself because I'm curious.
Is there some knowledge? Maybe somebody did a comprehensive, systematic testing on how does this shift of circadian rhythm affect the mice compared to mice that have a regular circadian rhythm as they do? Or is there just no difference there? Or do we just not know?
I think there is some limited research on that. But again, because most mice are obviously held in a laboratory environment, like you don't go out into the field and kind of try and track them. There is like alterations when you kind of mess with their laboratory induced circadian rhythm. If you have them like if you have like mice on reverse cycles, and obviously in 2 different rooms, they will exhibit different different traits, and obviously their hormonal levels will not exactly be reversed, but they'll kind of be messed up for some more so than for others. And also if you have them on like an unnatural rhythm where you have a 6-hour light and 18-hour dark cycle, that will also influence them. But one of the more interesting things is the few occasions where they actually go out and actually have wild type mice. Not Black Six that we bought that were unaltered, but actually go out and collect some mice out from the woods and bring them into a lab environment. Now, there's 1,001 thing you have to consider. They're stressed out of their minds because now they're captured, now they're in a cage. But in that case, the observations that I've seen at least, which again are very limited because that's not something I took a deep dive into, you get a significant shift and they don't feel great at all, which is not very surprising. But then again, we also have a cohort at the University of Vienna, which is being built up right now as part of the Shift to Health, I believe, project, which is examining these exact effects on humans. So the cohort is basically picking up people who are third shift workers, because their nutritional habits and their entire hormonal system is completely shifted. One might say completely out of whack, because, well, their night cycle is their light cycle. And when they work, they're actually eating while I'm sleeping, which again, one or the other is normal. Still kind of debating on that, which one it is. And even though we're very adaptable as humans, there are some certain constraints we can't necessarily get out of. Like the best example would be like getting enough sleep. Despite some people claiming they function on X amount or Y amount, like if you can function on that, you're the exception, not the rule. So even with nutrition, we're kind of working in certain constraints, and we're working with, well, a light cycle, which again gets more complicated as we move into the Scandinavian region where, well, for half a year, you're kind of basically messed up. And that's why we also see differences, mainly in vitamin D levels, also some hormonal changes, and also like huge rates of seasonal depressions because of a lack of light.
I wonder if there's other factors that can actually affect this circadian expectation, let's say, in mice. So obviously none of them see any proper light. They're all in the lab, so they're fully controlled. These are fully controlled conditions of, temperature and everything else, light included. But I wonder if you have mice that are controlled in the lab, one group being reversed for our convenience, so we can feed them when we want, so they're not sleeping when we want to feed them. But the other group having also light controlled conditions, but in their natural nocturnal schedule. I wonder if those two would have differences, if they can kind of feel, even though I'm seeing the light of the lab, and that is controlling my circadian clock, I wonder if there's something outside of that they can feel. It's not actually proper. This is not actually day. It's supposed to be night. I wonder if they can feel that, and if that can be reflected somehow. I don't know if there's any research on that.
I'm not sure. Again, we did not do it for ourselves in that capacity. I know there's some labs that do it. But again, going back to actually collecting wild mice from the environment, from the woods, they can definitely feel it because they know that even though it is dark in their environment, like in their artificially induced night cycle, that it is day. And the clock rhythm, and as well as some other genetical factors, are ingrained into our genetic code. So the circadian rhythm, when we started discussing it, and when we were on the grasp of kind of figuring out what this thing is, there was a lot of speculation. Right now, there is concrete data, and I want to say we pretty much know that our bodies, not just mice, but also humans, can definitely feel when there's something off in our rhythm. I think a good example might also be jet lag. So even though you're kind of switching from going into a region where you're offset by 8 hours, so you should be asleep at your current time, and also kind of more so adjusting to the light. Light is a huge factor, but one thing that I also figured out, or saw over the past couple of years of trying to follow Formula One a bit more extensively, is when they offset their natural rhythm as much as they do, when they go into a region where there's like a 12-hour difference to their normal cycle, they don't expose themselves to light. So basically they go back into like this lab environment, this synthetic environment, put the blinds on, tape over the windows, and just function on their normal rhythm. Because first of all, they can't adjust as fast. And secondly, if they try and adjust, they'll feel off. So again, that's mainly anecdotal, but I believe there is at least some concrete evidence on that. But someone else out there will definitely know more on that than I do.
So you're saying this is about Formula One drivers? Did I hear that correctly?
Yeah.
So that's very interesting. I would assume they need to really take care of it, because for them, even a split-second reaction time being off a little bit, which obviously you get with jet lag, that can mess up your whole career. Okay. So how does the timeline look like with I wanted to talk about other things, but this is interesting, so let's just talk about it.
Sure thing.
How does the timeline look like in the studies, such as in the one that you conducted with restrictive diets and mice? So first you have to, multiply your mice, right? And then you have to induce the colitis, and then you have to feed them different things at different time points for different lengths of time, and then see some kind of results. So how does that look like from the beginning to the end? Give me a timeline on that.
In our case, we didn't breed them in-house. We actually bought all of them because our breeding facility and our breeding capacities were somewhat limited at that time. So that's something we did not have to account for. Like, we got all of them from France, from Janivier, or however you pronounce it. I'm not a French guy. I have no clue how that's pronounced to this day.
Me here looking at you from the French part of Switzerland, a French-speaking part.
So correct me, please do teach me the ways.
Please don't ask me to teach you French. Okay, so go ahead.
And we bought them, and after we buy them, obviously we're if they're bred, they have like a period where they get used to a new environment. Like it's usually a week up until 2 weeks. Like the acclimatization period, we put them into cages. We don't do anything. We don't randomize yet. Just feed them like normal, just so they don't get completely stressed out. And especially if we're like dealing with inflammatory markers, which is the case for our experiment, you don't want them to be stressed because everything's going to be, well, affected by that. After that, we go into the randomization and obviously just split them randomly into groups. Usually done by software, and you just mark them, put them into a certain cage. In our case, most of the mice were held in group cages. I think we had to split the caloric restriction groups at some point into smaller groups because they get more aggressive. Because they obviously were the one group that's been most restricted, so we had to split them, but that's not relevant to the timeline, just kind of as a side note. And then we do our induction phase of the colitis. In our case, we did it through drinking water. So we mix it into drinking water. It was a 5% solution, and we give an acute dose of 5%, but it's done across like 7 days. So they're just drinking this DSS water. And after that, they obviously tend to already have the symptoms. We check for symptoms. We check the disease activity index, which is basically stool consistency, blood in stool, weight loss, and also just some patterns of their behavior. Are they getting more?
Can you, to any degree, control how much water, this colitis-inducing water, they are drinking? Is it just by giving them a certain amount that they will, in the end, drink all of it? Or do you actually track like how many sips they took?
We don't necessarily track it, but we can control it. Like if there's our bottles were usually 250 milliliters, and for the acute phase, we didn't necessarily like fill them completely up. It was like kind of half a bottle, I want to say. So we did not want them to drink the entire bottle because that would have been too much DSS, and the extent of colitis would have been too much for our needs. And after we kind of have a certain amount of inflammation, a certain, one might say, amount of colitis, we go into a chronic phase where we kind of maintain the phenotype that we have. So in our case, it was like continuing with 4 days on and 3 days off, if I recall correctly. So on 4 days of the week, they would again get DSS water, and on 3 days, they would get normal water by just switching out the bottles. So you'd have 2 bottles next to the cage, you just switch them around. Hopefully you don't mess it up. If you do, we're definitely going to notice because they also recover somewhat quickly because of their high turnover rate and everything else. And that's done throughout the experiment. So we have a week of them adapting to the environment, a week of acute colitis induction where we check for the phenotype, and then we maintain the phenotype for the length of the experiment. In our case, that was 6 weeks. And most of our experiments, and funny enough, most of like nutritional experiments are done across the span of 6 weeks.
What do you mean by maintaining the phenotype? You still keep feeding them the same water, just a little bit less, or okay?
Yeah, we do because again, in our case, there were black 6 mice. They have a genetic predisposition for colitis, but they don't develop it spontaneously. Like they are interleukin 10 deficient mice, interleukin 23 deficient mice that will spontaneously develop colitis, but it's like 1 in 8. But with our case, it was chemically induced through DSS. So you basically have to maintain it because, again, they're healthy animals, and the mechanism of colitis is just disrupting the colon barrier and allowing infiltration of bacteria. It's technically an endotoxin for the epithelial cells. And when you destroy the barrier, you get an influx of bacteria, you get higher inflammation, and it's usually just limited to the colon. Funny enough, colitis or DSS induces colitis through just disrupting the colon, and it leaves the small intestine basically intact, which is kind of interesting. Although the mechanism of DSS and the mechanism of action is not fully understood yet, we know that it's limited to the colon. So yeah, again, you have to keep feeding them or keep giving them DSS within their drinking water or some other methods. Like you could gavage them. You couldn't necessarily mix them with their food because it's a powder in our case, or it's 99% of powder, which is water soluble. And I'm sure they would kind of find ways to emit it while eating their food, even if you kind of mix it in. But yeah, maintaining it is also necessary for having certain effects at the end of the experiment.
And then at what point does the restrictive diet come into the point?
So basically 2 weeks after. So we have the 1 week of them getting used to the environment, 1 week of inducing more severe colitis, and then after that we have the diet, and the dietary intervention, as I mentioned, is usually 6 weeks, or in our case was 6 weeks. And over the span of those 6 weeks, again, they're fed either a caloric restriction diet, which is, I should mention, a normal diet that they would usually eat, like a standard chow for mice. It's just restricted in the amount. Intermittent fasting is also a standard chow diet, just restricted within a certain time frame. Fasting mimicking, as mentioned at the start, is like a mishmash of 1,000 different things that you kind of have to put together. And a keto diet is a special style or a special type of chow that was, like, I want to say, custom made for certain experiments for us, as well as for another partner group. And there were two different kinds of keto diets, which were differing in their fatty acid content. So it was either just long chain fatty acids or a mixture of long chain and medium chain fatty acids.
It's interesting that you mentioned the keto diet. I remember when I was in my master's in Heidelberg, we had an assignment to write a review, and the topics were already offered by different professors. So whoever offered the topic is the professor who's going to grade you, and they usually obviously give you a topic that they are an expert in. So I got the topic of keto diet, and I wanted to kind of approach it from a historical point of view. And if I remember correctly, the first human kind of study that was done with keto diet, it was a French study, and it was written in French. There was no translation. I think early 1900, 1905 or something like that. And I found it very funny to see one sentence about limitations of the study that I believe it was in a hospital, like a mental hospital. It was done to treat different disorders there. And one of the main limitations of the study was some patients were caught, sneaking into the fridge and eating, so they couldn't control that. So I guess you don't have that problem with mice.
Yeah, we don't. We don't. But that's always like a limitation of the keto diet, or matter of fact, any restricted diet, which is also a limitation for our study. Restrictive diets, as the name suggests, are restrictive, and there's going to be some adaptation, and there's going to be some willpower involved with it. So you kind of have to, I don't want to say force yourself to stick to the diet, but it's kind of forcing yourself to stick to the diet. And that's also, if we look into the beauty world and all these fancy diets, it all boils down to the caloric deficit. It's just how you achieve it. And if it's too much, you're going to end up binging. You're going to end up cleaning out the fridge within like a matter of minutes. So in that case, with the keto diet, one might argue that it's not as severe because you technically get to eat the amount of food you'd like. You just omit carbohydrates. But on the other hand, like, carbohydrates are like great for a lot of things, taste great, are usually in the foods we crave. So if you tell someone, hey, don't eat donuts because they're not keto friendly, guess what they're going to eat? Donuts. Guess what they're going to crave? Donuts. So anytime there's like someone in the vicinity of like 3 nautical miles, they're going to smell that donut just because they crave it so much.
If you mention a donut one more time, I might have to leave this podcast and have a donut. Okay. So, and then what do you measure in the end? Okay, so you say inflammation. I guess there's just a bunch of inflammation markers. That's some kind of genes going up, or some kind of products of the inflammatory system just going up in certain parts of the body. How do you in the end evaluate what happened to this mouse during a restrictive diet? In your case, at least.
In our case, we weren't as like thorough methodologically because we did not have the facilities to do so. So it boiled down to Western blots, qPCRs, cytokine arrays or protein arrays, I should say, some HPLC measurements, and so on. So as you mentioned, we definitely look at gene up and down regulation because it is believed that colitis is definitely in part due to genetics, as are like all of these complex inflammatory diseases. So we looked at like transcription factors for different cytokines, starting from all of the interleukins, all of the type 1, all of the type 2s, then going into NOD2, which is usually like one gene that we can universally agree on is related to inflammatory bowel disease, more so Crohn's disease than colitis. Then we move into checking for protein levels, absolute differences via Western blots, relative differences mainly through the arrays, which are basically just modified ELISA. Technically, it's just the same technique as the ELISA, just that you do it on a small slide instead of like the well. And during those measurements, we also did a huge amount of cytokines. We did 40, anything from like your usual suspects of TNF alpha, TNF beta, interferon gamma, all the way to GM-CSF, GMSF, and so on and so forth, looking at as many things as possible because this has never been done before. So we kind of had a shotgun approach in that case because we didn't necessarily know what to look for. So we looked out for everything that could possibly be related and just went from there. So when we got our results, then the interpretations were based on the results necessarily, or not necessarily the other way around. Because again, we did not have a lot of literature to base our primary experiments off of, but we did them and then kind of based on the results of that experiment, continued further down the path. So we were kind of our own reference in that sense, which again is not the greatest starting point, but there was no alternative in that case.
So when you say that some of these restrictive diets have a positive impact on the mouse, are we just talking about bringing these genes back to normal and protein levels back to normal, or do you also examine something like, okay, if it had poop in the stool, does it have less? If it had blood in the stool, does it have less blood? Or behaviorally, is the mouse just doing better? What are the parameters to say that this diet is now good for this mouse or better?
I'm glad that you mentioned it because that's something I usually forget. I get caught up in the technical things because the things you just mentioned are like the basics of what we actually look for before actually analyzing the mouse while it's still living. And that's also a criteria for human subjects. I mentioned it at the start, the disease activity index. Like there are different indexes and different kind of ways to classify the extent of the disease, but it's usually based on weight loss, behavioral changes, blood and stool. So after we induced the diet or after we started the dietary intervention, we generally saw a slight reduction, although not significant, in the disease activity index. So their weight stabilized, they had less blood and stool, they had less frequent bowel movements. Yet, funny enough, the keto diets actually reduced the weight even further. Which, if I recall correctly, I hope I do. If I don't, I guess I'll just correct myself later down the road in the paper and just be like, whoops, messed up. But
I'll let you know if any angry nutritionists are.
Appreciate it. But yeah, we kind of indirectly saw that weight loss isn't necessarily the best factor to base this off of, because again, we saw a further decrease in weight loss, which would usually indicate that the disease and that the colitis is more severe. And when we analyzed inflammatory markers later down the road after sacrificing the animals, we saw, oh, these animals, despite losing a lot of more weight, were actually, well, less inflamed, to put it bluntly. So there's definitely things you can measure. There's also dextrin scans or dextrin experiments, where you basically measure the permeability of their membranes, be it intestinal or be it colonal, where you gavage a fluorescent dye with dextrin in it. So it permeates or it gets absorbed into the bloodstream. And then you take off blood from usually the tail vein and you measure the fluorescence of that. The more you get, the less integral their barrier is, meaning, well, we technically have more colitis. Again, very indirect, but it's used in nutrition quite extensively because we're usually looking at, first of all, what permeates into the blood, and secondly, how much of it gets into the blood. So if we have a membrane that is perfectly intact, well, we don't have to worry about it. It might get absorbed, but not to a pathological extent. But if you mess it up with certain chemicals, like in our case, dextrin sodium sulfate, well, this membrane is not as fixed, not as integral as it used to be, not as tight as it used to be, and you're going to get a lot more infiltration. And if this foreign substance that we introduced got into the bloodstream, there's a huge chance that endotoxins, other bacteria, and everything else also got into the bloodstream and are indirectly causing all of these problems. So there are screening methods during it. You could also do x-rays of the animals to see their body composition. We had the capacity to do that. We did not do it for time and budgetary constraints. But there's definitely things you can do to kind of track the animals while they're still alive.
It's very interesting. I'm not familiar, or I wasn't until now, familiar with how do you test permeability of membranes in mice. We have I work with flies, as you know. And in the fly field, there's one test, one assay that a scientist called Michael Herrera from France developed. It's called the Smurf assay, where you feed this blue dye. You put the blue dye in the food, and then basically if the flies are having permeable intestinal barrier, it just turns blue, and that's why it's called the Smurf assay. So kind of the proportion of these Smurfs you have. And then it's kind of debated of how good the assay is. It's a little complicated how to interpret the results, and people are always fighting about it. I wondered, is this the only assay that's used with mice for permeability? Or because I know this Smurf assay was also used with zebrafish and maybe some other animals. I don't know, is it with mice? Are you familiar?
As far as I'm aware, this is the first time I'm hearing of the Smurf assay.
Okay.
So I no
idea.
I guess not.
All right.
I guess not.
All right. So it's an interesting assay. At least that the name is interesting and the phenotype is interesting. Quite binary. You see the fly going completely blue. It's a Smurf fly. Okay.
So yeah, before we move on, just one more thing, which is also something we did and is usually done again in nutrition-related research, which is a gut transition time or transient time assay, where you basically feed them a dye. Usually it's Carmen red, and you basically wait for the mice to poop. So you're just sitting there and just waiting for the red poop to appear, which is a ton of fun. It's not. But in that case, you can kind of look at the transition time from mouth to anus, or from the moment they ingest something to the moment they excrete something. And also that can kind of give you insight of what's happening inside. If it's super quick, well, we have basically diarrhea, we have a lot of fluid buildup, we have a lot of fluid reabsorption into the stool, and so on and so forth. If it's too long, we might have a lack of fiber, we might have some other issues. And if it's normal, well, then it's normal, and you just spend like 4 hours or 5 hours watching a mouse poop. Great. Welcome to science.
So if you had a band, it would be called Pink Floyd.
Yeah, exactly.
Right, sorry, I cannot resist a bad pun. Okay, so congrats on the paper. Maybe let's switch a little bit to talking about the process of writing a paper as a master's student. It's not the most common thing in the world to be in a position to publish a paper as a master's student, so it's a good achievement. But I guess it's also stressful to do it at that level, that early in your career. So maybe if you can tell me a little bit about the process, like what were some challenges, what was difficult about writing the paper, maybe interpreting the results? Just guide me through the whole process. How was it for you?
No, it wasn't as stressful for me, but then again, I want to say I was somewhat decent at managing it. And the entire goal when I got into the lab, which was like April 2021 or something along those lines, or 2022, I can't even remember. Like the last 3 years are kind of a blur, which I think a lot of people will relate to. But the goal was to learn. And I like to say that the paper was kind of the cherry on top of my learning process and my development process and my growth as a young researcher, which I'm still completely in, and it's still a road to becoming an actual researcher. I'm still very cautious of calling myself a researcher, so to make that clear. But it was kind of weird because I got to the point where I finished all my results. We did some revisiting, and when I say some revisiting, I did like an additional 60 qPCRs, which was very fun. On, of course, 384 well plates, which was amazing. I love them. I still keep them somewhere in the lab unless someone threw them out. I'm gonna pick them up at some point.
Do you also have do you have a good method of tracking whether you pipetted a certain well or not? Because for me, it's always I start thinking about something during pipetting and I forget what did I pipette and what I didn't.
Funny enough, I actually do because I have found a setup, and again, it's going to be a digression from the topic, but we'll get back to it. I found a setup in our lab which was good for me, and it really worked out. We did everything in triplicates. So first of all, I would split the plate into like these 3 well parts, and then I would put the well or the plate on ice because I noticed that those 2 microliters, by the point I get to the last well, evaporate. And I put it on ice, and I actually put it on our dissection table and turn on the light from underneath. So every time I would pipette, I would have this drop, and if you turn it just perfectly, you would see the shadow of the drop. Like you wouldn't necessarily see the drop, but you would see kind of something lurking in the well. So even if I kind of messed up or started talking with someone, I just look over, see which wells look darker than the others, and I know where I was. So that took me a lot of time to figure out, and that's a very specific setup to our lab. But if you can kind of find the perfect lighting and the perfect angle, you kind of can see the actual 2 microliters in the well. Now, if you're pipetting 1 microliter, everyone be on your side. Like it's probably going to be impossible, but yeah, there was a way for me to kind of keep track of all those wells and not get messed up as many times. I still messed up quite a few times.
Well, good that you found the way that works for you. For me, it's always a struggle with attention whenever it's something with so many details. So going back to the paper and the whole process, you said you found a good way to manage the stress, and you were fairly decent at it. Tell me, how was it from the beginning to the end?
Well, I basically finished up everything and wrote my master thesis. And my supervisor, who is, well, technically still my supervisor, always kind of made fun of me that I have a very specific style of writing. So my thesis basically reads like a paper as well. So it's not like, one might say, as relaxed as a thesis might be. So it was kind of easy to take the material already written and adjust it slightly to fit a specific journal. In our case, it was the Journal of Nutrition. And in that case, it was like, okay, I wrote this, and now I basically have to write the same thing, but slightly differently. So I ended up paraphrasing myself. So I took my master thesis out and went through the entire thing and kind of wrote once again. And obviously not as extensively, although my master thesis wasn't all that extensive either. It was like 50-ish pages, even though there's like an unofficial requirement to write like 70 or whatever. My supervisor was super happy that it was short. Let's put it that way. But basically, the one thing I struggled with, and the one thing that ended up being so stressful that I actually had to manage, was the discussion part. Because again, we're technically like one of the first labs doing this. There's there were two papers out on the keto diet in colitis in mice, both from China at that point, and there was nothing. So you're kind of analyzing and discussing on a rudimentary molecular level. So if you see a difference in a certain factor, be it TNF alpha, again, taking that as a prime example because that's one of the more consistent factors when it comes to inflammation, you look at something like a similar disease, another inflammatory disease, be it multiple sclerosis, be it rheumatoid arthritis, be it anything else that has a TNF alpha component. Because again, taking colitis into consideration, there's not going to be a lot of data. And then you also take clinical data from cohorts. You look at the levels in certain patients. You look how the levels change because you can't necessarily find something that will be one-to-one like the stuff you did because it's not. But I think that's kind of the struggle you generally have with research. If it's not publishing worthy, that it's novel and you're kind of the first one doing it. So managing that was kind of stressful. But the one thing I had as an advantage is a very good supervisor. She didn't push me, and it was like, "Would you like to publish this?" And I'm like, "Sure. If there's a possibility to do so, I'd like to do it." There wasn't any point where she said, "Yeah, we must publish this," or "You must publish this," or whatever. It's her data, her offering me to actually publish and be the first author. Well, to be honest, no one else did as much work, or basically I was the only one doing this project. But then again, if I said no, I would have been omitted from the project in the paper itself. But that was one thing, and the other thing was like I knew that at no point I wanted to push myself over the limit, and that goes for a lot of things. If it stops being fun, I stop usually doing it. And it has to stay fun to a certain extent. Sure, there's going to be days where you're going to wake up and you're going to hate your life and you're going to hate your job and you're going to hate what you do. Then you eat breakfast, drink a coffee, and it's better.
You learn to have fun in the hate.
Yeah, but yeah, I mean, it sounds very like black and white, but I really didn't want to get to a point where I started hating science. So I took like a casual break where I didn't do anything for a week. I took a break where I didn't write. I took a break where I didn't feel like doing experiments and so on and so forth. And again, I had the opportunity to do so because of my supervisor. I know a lot of people who were not as fortunate to be in such a situation where they had a supervisor who's understanding and full of compassion and actually giving you support. So I can't credit myself for managing this as well as I did because like at least it's 50-50 between her and me and us managing the situation together because she understood that I'm just a master's student. And even when there was criticism, it was criticism in regards to me being a master's student. I wasn't expected to perform at a postdoctoral level because I'm not at that point. I held myself to a higher standard, which I always tell.
Kind of toned down criticism, taking in consideration that you're just starting out. Well, I think that speaks a lot to how important it is to find a supervisor who can do that, because not many people are that empathic and understanding and able to give you a critique that is constructive and not offensive, and take in consideration, all of the factors that, you're just starting out. This is your first, maybe your second research project, first time gathering so much data to, that it can be publishable. So can you speak to me a little bit about, were you just lucky to get such a good supervisor, or was there a process behind finding the lab that you ended up in? And could you share some of the wisdom on how to find a supervisor like that?
Well, to be fair, that was like pure luck in that case. Because at that point, I didn't think much of it because I just wanted to get into the lab. It was at a funny time because I was still in my bachelor's, and given COVID and all the restrictions, the last 2 exams I had to do for my bachelor's were split across like a year. And I thought to myself, oh, might as well do something useful. And that's when I saw a post. I don't even remember where it was. I think it was not like one of our Facebook groups, one of the many Facebook groups for our department that are like dead. Like there's one post per year. And I just found her email and I just wrote her. And we started talking, and there was a lot of confusion of what I wanted to do because at the time she was actually looking for a master's student to do a thesis project. And I was like, can I get into the lab just to do some lab work, just to do a voluntary internship at that point? And then at the point where we got onto the same page, we kind of started discussing what I would do for the first couple of months and so on and so forth. I followed and I shadowed the other students. I helped out around the lab. I got to know the lab. Like,
Wait, so was this a required internship, or was it like
Nope.
really voluntary,
Yeah.
outside of your regular curricular activities? Okay, so you reached out. So what you're saying to me is you reached out, you asked to contribute or try to learn outside of your regular schedule, and how many months later you actually get a paper out of it, which I think speaks a lot to that proactivity, right? So tell me more. Tell me more.
Yeah, because it's definitely like the $1 million story from going from a plate washer to a millionaire, like the typical American dream kind of thing, where I started really from the bottom. Now we're here. Well, I can stop myself from doing that either. But I was literally just getting to know the lab when I started. Like, where the hell did the pipette? Where the hell is a pipette? Where the hell are the tubes, and so on and so forth. And after a while, I finished my exams and I got into the master's. And then at that point, it was kind of like, do you want to do your master thesis with us? I'm like, sure, why not? So at that point, I kind of started off, I might say, on the wrong foot. Turns out that might have been the best thing that ever happened to me, because I had my master thesis basically done before I did a single exam and a single lab for my master's. So that was kind of like weird, and is still to this day weird. But in the process, I kind of learned everything by myself. And then when I actually entered the labs and the exams and everything else, I was like, oh, I already know this because I taught it myself. And I taught myself, which is kind of crazy. But yeah, going from just doing it on a voluntary basis to being offered the master thesis position to actually doing my mandatory then afterwards, because during my master's I also had a mandatory internship. I also basically finished up that as well during my time in the lab, to publishing. I want to say it was like, I think I started in April 2021. No, it should have been April 2021. I think it is.
Okay.
So roughly 2.5, almost 3 years, or not 3 years, but 2.5 years later, we got the first publication, and it was finally published in actually January 2024. So I guess 3 years later down the road, we might have the second publication out of that as well. But the proactivity that you mentioned is the thing that you talk about on your LinkedIn, that I talk about on my LinkedIn, that so many people talk about. And it's been so cliche to hear it so many times. Like, to understand people getting frustrated, but it's a thing they got to do. Like, you have to take your own future into your own hands and kind of take responsibility for what's going to happen to you. Because I got the opportunity to study in Vienna. That's definitely an opportunity that was presented to me by my parents, by my family. Sure, I had a wish to go to Vienna. I had a wish to do my bachelor's here. But if they didn't pull through, obviously it would have been impossible from a financial point of view and everything else. And then afterwards, I started using all the resources and started using them to the best of my ability to create opportunities for myself. Because again, the moment my supervisor was looking for someone, she was looking for a master thesis project or a master thesis student. I wasn't that person. I was still in my bachelor's. I just wanted to do some lab stuff. I just thought the lab was cool. And later down the road, I busted my ass and started working and did as much as I can to actually, well, get to the point where I could feel comfortable in the lab. And funny enough, I do it to this day. If I see like an internship kind of thing or someone looking for a master's student, even though I might not meet the criteria, I'll likely apply and who knows? I'll explain my situation. And if they're willing to take me given the circumstances, yeah, sure, why not? So proactivity is the name of the game. And yeah, you just got to do it. So
How is it to study in Vienna? So you said, this approach of proactivity, you approached the professor, you wanted to learn, you offered yourself, your help, and whatever you could offer as a bachelor student who still had no significant experience in the lab. But I know from the time, because there was a time when I lived in Vienna a long time ago, and I kind of know the vibe of student life there. I know many people study plus work at the same time, so they kind of part-time study, part-time work. Can you tell me a little bit about your experience and how is it just to be at the University of Vienna? How is it to study there? Do you have to work part-time to kind of sustain yourself? Just how is it?
well, I got to premise it by saying that my life is very specific in a way that it suits me very well, and I like it. And it would look very weird from a lot of people looking from the outside in. I mentioned my sports background, and through my sports background, first of all, I got to be very independent at a young age because training camps, training, studying, going to school, and kind of having a good chunk of time outside of school allocated to training was already like an extra chore that most people didn't have. So at our peaks, we used to train 8 hours per day. So go figure out how much free time we had. But that thing kind of carried over into my adult life or my post high school life, where I was officially an adult with 18 years down the road and everything else. And I knew from the first moment that Vienna is kind of going to be a station, not necessarily a destination. Because I knew I want to do my degree here. I want to do potentially, well, it ended up being my master's as well. And I want to go further. I want to move to a certain place. I want to experience different culture and so on. But you mentioned it yourself. Vienna is very specific. First of all, yeah, it is the most livable city in the world for the past who knows how many years. There's probably the coldest city from a people point of view that one could imagine, or at least out of the cities I visited. People are not necessarily like outgoing. People are not necessarily friendly. So get used to maybe being an outsider, especially if you're a foreigner, especially if you don't know the language. I want to say it's at points even worse if you know the language and have a heavy accent because you're going to be judged to hell. And again, I was lucky enough to have grown up speaking 3 languages, so that was a huge plus because, well, German is technically my second native language. But working and studying was hard, and that was also a potential, well, source of stress. And in most cases, like, unless you have like huge funds, it'll probably be necessary, especially like for the past couple of years with the economy going belly side up. You can definitely see a huge difference and a huge shift. Like student life is not going to be spectacular, and that's one thing I want to also premise or also want to point out to most people. Like when you see students doing these things and students doing fun things and doing research and whatnot. I'll take myself as an example. It looks great. I published. It is great, but there's
Then you
so
go home
many.
and sleep on the floor.
Yeah, but there's like so many unfabulous things behind that one has to account for and one has to be ready for. So if you go through Instagram and look at all the fun reels of all the fun places in Vienna, yeah, they're definitely fun. There's the main library, which I like never visited because I can't study in the library. I just can't get myself to. It is Hogwarts like. You will enjoy it. There's a lot of things to see. There's the centrality of Vienna and the connection to other cities like Bratislava and Budapest and whatnot, so you can take field trips on a weekly basis if you're that kind of person. If you have the time. The one thing that's again has to be considered is it's a student life. Like, you're trying to make money, you're trying to squeeze through, you're not living a glamorous life. And anyone who does, there's some external funds flowing in. Like, we can go through my finances from top to bottom. I'm always open to talking about that. And I can already tell you, it's like every single month, especially for the last like 5 or 6 months, I break roughly even, where I have like 100, 150 euros to spare. So there's some kind of comfort in a sense that, yeah, I don't have to worry about like my living expenses, whatnot, my having a roof over my head and having groceries and everything else. But again, those are the bare necessities. And that's the type of life I've been living for the past however many years. So in that case, it's not stressful for me, or it's not depriving myself of certain things. But for someone else who's used to going out and everything else and partying and pouring a lot of money into a certain type of lifestyle, it's probably not going to be great. Unless, again, you have someone on the side or something on the side that's sustaining you in a very meaningful way. Because you can throw out 500-600 euros in 2 weekends.
Mm.
And, well,
Are there any opportunities in Vienna to get a paid internship in a lab that has a lot of funding, or in a company that might hopefully use your expertise that you initially acquire, and then later down the road you have some kind of contract? So are there opportunities in Vienna and maybe broader Austria to get paid for what you're doing in the lab, along with, let's say, if it's a master thesis or an internship?
There definitely are. And again, I had the opportunity to also work part-time as part of the University of Vienna, as part of the Medical University of Vienna. Like, those were paid positions, but those weren't necessarily internships. Those were like lab tech positions and tutoring positions and so on. But there are also internships that are paid. And internships are like compensated either on a part-time basis, which ends up being 1,200 euros per month, which is very livable. Again, talking from experience, that's about the amount of money I take home on a regular basis as well, on a monthly basis. And there are also certain internships that are paid as a stipend. So in that case, it's going to be about 500 euros. And it depends on the project. As always, like, go through the fine print and look into it. And in Vienna, there are a lot of opportunities. Like, you have the main university, you have the medical university, you have the SEM, which is the medical technology center or whatever, which is kind of a gray area between academia and the industry. And although I usually find gray areas kind of weird and they don't work, in this case, it's definitely the best like research facility in the entirety of Austria, hands down. And funny enough, they're looking right now for like 2 student helpers, which are compensated, and 3 master positions or 3 master thesis positions, which are also paid. So there's opportunities if you look. There's the Vienna Virus Center, which is like 500 meters that way, which I did not get the chance to explore yet. Still looking into it, which will be very useful because it's right in front of my door. And there's around 50-ish labs, anything from startups to types like Thermo Fisher. And again, it might not be as easy to find. There's a lot of stuff online, but as we know, there's a lot of stuff also being spread by word of mouth and just, I hate to use the word, but networking. Hate to break it, but it's like, if you talk to your fellow students, if you talk to your fellow lab members, PIs, lab managers, and again, lab managers are probably the saints of the entirety of the academic industry and probably the most knowledgeable people you'll ever encounter. They'll, first of all, know all the gossip of the institute, and secondly, know everything that's going on within the institute as well as outside the institute, and will be the first person who's going to have a lot of knowledge and a lot of useful knowledge for you. So talk to the lab managers whenever you get the chance, if you're applying to jobs or anything else, because they know a lot of stuff. But there's definitely opportunities. Again, you have to know where to look for. You might have to know how to look for them, but there's a lot of them.
What about, okay, so that's the situation if you're already in Vienna and it's already there, you can talk to people. What if I am now in India, or what if I am in Bosnia and I would like to apply to an internship or a master thesis or something like that in Vienna? Is it a common practice? At least, have you seen this happen that labs will take international students, even if it's just for a short time?
It is common practice, and again, it kind of depends on the type of position. It is less common within the universities because, as we both know, universities are very specific in a way that even though a position is advertised, it might already be taken. But let's not go into that. That's going to be a rabbit hole for another time and another place. But for institutes, again, like the CEM and like the Vienna Biocenter and like all these other facilities that are not directly linked to an academic environment and an academic facility, they tend to actually prefer international students from what I've seen. Like the CEM, a few positions I wanted to apply for, and never got the chance to. I looked later into the lab, and I looked into the group and into the group members, and they recruiting like people from Mexico, from Canada, from China, from Japan, and so on and so forth. So there is opportunity. Again, it's going to be a lot harder for them, but with institutes, it's a lot more common and it's a lot easier because they'll cover like your expenses for the interview and stuff like that. Whereas the traditional academic system, like the University of Vienna, if they're looking for master students, it's going to be usually a master student recruited within the group or within the department, or to say the least, from Vienna, because they won't have the necessary funds to actually get someone out from an XY country, get them the financial support needed, get them the administrative support they'll need, get them the bureaucratic support they'll need, and so on and so forth. So in that case, I will definitely recommend looking at institutes, just because there's higher chance that you will get something out of that rather than the universities.
That's pretty cool. A common problem is, when you're trying as a biomedical early career scientist to find a job in a company, it's usually this hassle of they won't sponsor my visa, and it's difficult for them to take me. So why wouldn't they just take someone who's right next door, who's already living there? But I guess in the academic setting, it might be a little different in case they have funding for those kinds of things already secured by the government, or something like that. So would you recommend for students to reach out directly to professors, or if they are living abroad and trying to get to Vienna or Austria in general, or how would you recommend that they apply?
Like reaching out to the professor, you cover this in more ways than one. First, let's go to your LinkedIn profile and read for all the tips and tricks on how to approach a professor and what's all the prep work to actually make your time worth their time, and then actually start looking for people to apply to. Yeah, applying to professors should be the pinnacle of it all. Try and get to the, I want to say, lower ranked people, if like the PhD students, the master students, the people who are, yeah, basically the peasants of the academic world.
Peasants.
Yeah, but get talking to the people who are in the position that you are in. Like, again, find a cliche, but ideally you also find someone with a similar background. Like if I were looking for a position in, let's say, Australia within a certain institute, I look at someone who has the same citizenship as I do, because it's likely that they'll have the same administrative and bureaucratic struggles that I will have, because I'm not going to be a special snowflake. And talk to them and their experience is going to relate a lot more to me than someone who's coming from the US. The same goes for Europe. Like you already mentioned it, it's going to be easier for someone to hire locally. At every point in my career, when I was applying to something, I knew that basically I had to be like 20 or 30% better than the next local candidate for them to be worth their time. Because it's going to be a hassle for them to go through the entire application process with me. It's a sad reality. It is the way it is. So in that case, you've got to be ready to be better and a lot better than the locals. And getting back to your actual question, talk to or write to professors after you talk to PhDs, after you talk to master students, after you talk to postdocs, then write to them. There might also be positions that are already out there. Look for those. And also there might be open calls for certain things as well. Like the best example would be like the open calls that will be starting at the Medical University of Vienna for PhD positions, which will be starting at the 1st of March. So they have an open call, which is not uncommon, but like not the most common thing either. So in that case, you kind of throw it into the pot and hope for the best instead of like going the Swiss route where you have to apply directly to the professor, ask for a position, and so on and so forth. But again, that's kind of going into the finer details of the specific positions.
I think it's a great advice to contact people who are in a similar background as you are. And many times it happened to me that when I was exploring labs that I wanted to apply to for PhD or for an internship or master's thesis or whatever, I would contact the alumni, and actually I would change my whole position on the lab, because I would get first of all, how is the lab atmosphere? How was it to be in that lab? What are the policies around publishing and working, and how are they working, and how are they doing their lab meetings, and how is the mentoring and all of that? And how difficult is it actually to get there from this background? And, getting information from people who are just one step ahead of you and from a similar background as you can be tremendously useful. And, for me, LinkedIn was the platform that was the easiest one to find these people. Okay, cool. So I want to just go back a little bit to your writing process and paper publishing process as a student and working in the lab. Were there some, we talk about research, I feel, according to a certain cliche pattern, and we always say the same things. Could you maybe share with me some unexpected challenges you encountered? Something that, someone who hasn't done a research project thought, that's not going to be a problem. But then you start doing it and you go, okay, this might be a problem. I did not expect this.
There's definitely more than one. Like, I learned to hate Western blots. And the one thing
I said the not clichéic ones.
Yeah. But yeah, that's like the first thing that comes to mind. The second thing is definitely like things that usually work are qPCRs. And if you have like a standard routine, the protocol you have in the lab that works every single time, I'd say is an unexpected and a frustrating thing when it doesn't work. We had a situation with our qPCR for qPCRing some bacteria strains, and none of our primers worked. And the first thing is that, okay, we didn't have enough concentration of our starting template and just add more. Again, nope. Annealing temperature was wrong. Test it again, nope. And we couldn't get that working up until the end. And at the end, I wasn't the one who got it working. It was the lab tech in our lab.
Sorry to interrupt here. Just for the people who might not be necessarily biologists, just briefly maybe tell me, what is a qPCR?
So basically, a qPCR is the next development of the polymerase chain reaction, where we quantify the amount of genetic material that we started out with. So basically, you have our template, which is basically our DNA that we go into the reaction with, or we put it into a tube, or in our case, a plate. We add primers, which basically recognize the sequence we want to amplify, and then we go into the actual quantitative part, so the Q part of the PCR, where we put it into a so-called thermocycler, and then we basically get our readings of how many cycles it took until we multiply that starting material up to a point where it was detectable. And the detection is usually color-based or fluorescence-based. So anytime during COVID you heard like a CT value, that exactly talking about qPCR will reach a certain CT threshold and recognize the starting material.
So in essence, just whatever gene you're talking about, is it going up? Is the product being made, or is it not being made?
Yeah.
So just to, sorry to all the biologists that we had to mention primers and all the other stuff. So, okay. So yes, sorry, you were saying about the challenges.
And the lab tech managed to figure that out. And the second challenge was kind of planned, I want to say, planned by my supervisor, because at that point I was in the lab for quite a while. She saw that I was working. She saw that I was working quite independently. At some point, she wanted to do an MPO assay, which is myeloperoxidase, found in mainly white blood cells. In our case, we were targeting macrophages, which are like the resident good guys of tissues that eat up all the bad stuff that shouldn't be there. So we wanted to measure MPO, myeloperoxidase, and she basically gave me a paper and said, well, go figure out how we're going to do this essay, which was equally fun and equally frustrating because 3 months later, I had something that I thought might work. It was the most challenging experience in my life, but it was also the most fun experience because I had gained enough trust from her side that she could basically give me something and expect me to do it by myself. But going back to the challenging side, you get a paper and you read something, and now you got to replicate it. So the first thing you do is like run through the lab and figure out if you even have the raw chemicals. And then you go to other labs if you don't have them, and so on and so forth. So those are the two things when it comes to the actual lab work. And when it comes to the actual post-lab work, it was definitely like all the statistical stuff and dealing with the amount of data we had. Like with the protein array, analyzing 40 cytokines in quadruplicates, trying to normalize them based on a blank, trying to actually make sense of the data. That's like took 4 weeks of like nonstop work. And yeah, at the end, I got what I wanted. But that entire process, at some point, I was like, yeah, I'm just going to open a bakery. This science thing is not working out. No, like the standard contingency plan, just open up a bakery.
That's very funny because one of my friends who was studying genetics with me, he actually opened a bakery after. He's doing quite well, much better than most of us as biologists are, especially in our home country in Bosnia. All right, so the no gluten factory in Sarajevo, if anyone is interested. Okay, so I have this image of you. We have met in person once, I believe, right? It was just once in Sarajevo in that meetup of the BH Futures Foundation. But I more know you through LinkedIn and your posts, and you often share your tips about being more productive or not wanting to open a bakery, hopefully, if you're doing research. And I noticed many of your posts, once or twice a month, you will go and make a detailed report on what you did this month. So that involves, I guess, some things that you would call taking care of your mental health, like some physical activity, how many papers you've read. That's another interesting one, which I always get very surprised. Like, how the hell do you read so much? This guy's suspicious. He's not reading this many papers. Is this abstracts? You have to be a little bit more concrete about this. So you give this detailed report, and you very openly say, damn it, I planned to read this many papers, and I didn't finish it. I wanted to read 25. I read 12 because I was feeling like shit the whole month because whatever. And I think it's interesting to openly talk about setting goals and sometimes coming short of them. But also, I noticed you've been doing this for quite a while now. And I feel like the kilometers you're doing running are going up, and the number of papers is steadily going up. And all of these metrics you set for yourself are steadily going up, even if you're occasionally having a bad month. So maybe can you tell me a little bit about how did you start doing that, tracking your progress publicly, and how has that helped you or not helped you, and have you changed your psychological approach to how you see your progress and so on? Just maybe talk to me a little bit about that.
Yeah, I mean, I guess before we start, we talked this off-screen, we mentioned as a stats guy. I'll probably adapt that to my liking and use it as a way to describe myself because I really like it. But I guess I did this in some capacity throughout my entire life. And this is something I wanted to do back when I was like 14 or 15. I wanted to quantify what I actually do. And again, this might be like a consequence of doing more science where you actually want to graph stuff out and you want to have concrete data and you want to compare all those things. But the second motivation behind this was, what if I'm sharing the stuff I do and these tips and tricks, and if someone might expect, okay, he's doing all this great stuff, which is inspiring to some, boring to others. I want to make it more relatable in the sense that you can actually look through the stuff I did and the work it takes to get to that point. I want to demystify the entire process, because one of the things I always disliked is like these motivational reels on Instagram and whatnot on social media where you have this millionaire who at this point is completely unrelatable. Like, they might have started from a humble beginning, they might have worked their way up, but given that they don't connect with the average person, that they don't share like their everyday to-day life, but only the highlights, they're not relatable anymore. And I guess in some way I want to stay relatable, and I also want to keep myself accountable. And then something I took from sports and everything else, where you kind of share with the public and kind of be in the public eye for better or worse. It has helped me in more ways than one. The first thing is, like, as I said, I wanted to do this back when I was 14, 15. And at this point, I finally managed to do it. And at this point, it's like the second year going. So 2023, I started, and now it's 2024, and I'm still doing it. And I'm tracking myself when I go to sleep, when I wake up, the papers I read, the amount of time I spend of self-improvement, which is somewhat gone, like learning a new language, learning a new skill, and so on and so forth. And a couple of other things. I might add or I might subtract over the year, but it's generally that kind of backlog of things. And one thing that I noticed is, first off, it motivates me in the sense that I look at the previous month and I want to outdo myself. I was always results driven. Again, going back to my background as a swimmer, like that split second, not even a second, that split second, I knew that I had to work very hard to get it down, but it was very rewarding. So going from January into February into March and trying to read one more paper or do one more activity or do one more workout is also very rewarding. But the second thing it created is kind of a restart point, because every month is a self-contained unit. I do the month, and yeah, I look at it as a reference for the next month, but I kind of forget about it. It's like, yeah, that one was shitty, wasn't great. This month is a fresh start. So I start from zero, basically. And it's like this New Year's resolution kind of mindset every single month. I don't go into it like, oh, I was so bad in February, or I was so bad in January. I'm definitely going to be bad in March. No, it's a clean slate. Like, this entire month is entirely isolated from everything else, and I'm going to keep doing it, and I'm going to try and beat my previous best, and so on and so forth. And as you mentioned, it is kind of an uprising trend that I try to keep. And again, I'm being honest. If it's a bad month, it's a bad month. I'm still going to share it. I'm still going to put it out there, because that's normal.
I think the most important part there is, if I imagine myself doing this, or somebody else trying this for the first time, let me track everything about myself, everything that's important to me, the papers, the when I wake up, the when I sleep, the when I exercise, how many times I exercise. I guess the most difficult part in the beginning would be, oh shit, this month sucks, and I sucked. And then I guess there's a crossroad on how you're going to look at it. You can look at it as, I tried it the first month and I failed already, so why even try? Or that shift of like, okay, there's a reset. Let's just do it next month again, and let's try it again. And then you fail again. No, but how did you deal with it in the beginning? Was it as easy in the beginning to just hit reset and go, another month is now a new resolution, or was it difficult to get that momentum?
In the beginning, it was easy because, again, I started in 2023 and I started in January. So it was like, okay, I'm doing this new thing and I want to do it to the best of my ability. And the first month was like already quite decent. I tried to outdo myself in February, March, April, May. I think the one May actually almost completely fell apart. And then June and July, I was just like, for the majority, I was just on holiday and I wasn't doing anything. And that's also something that I tracked. I was like, I didn't do a single workout because I was traveling. I was just sleeping in. Like my average wake up time went from 6:30 to like 11 or whatever because I was just being lazy. But that's something I needed. I didn't have a proper vacation for many years. And at that point, I was like between jobs and I had 9 weeks of vacation or something crazy like that. I would say it was 8 or 9 weeks. And I completely used those 8 to 9 weeks. I didn't read a single paper in that period, or like 2-ish.
Naughty boy.
There wasn't like a lot of self-improvement going on, like I was learning new skills or whatever, but that's what I needed. And that kind of circles back to the stress management thing. I allowed myself to be lazy because I knew that it's a marathon, like it's an ultra marathon. It's not a sprint. Like I choose a career that's going to be fruitful after 20 years. I'm 27. I have like 20 years before I get into a position where I'll do truly my own research. It's going to take some time. And if I want to stay in this career and if I want to stay sane or remain as sane as I currently am, because we could argue that no scientist who chooses the path we do is completely sane, to put it nicely. I'm stumbling across the world because I'm trying to censor myself and try and be correct without being too aggressive. But yeah.
Always consider that nobody's going to listen to this podcast. Probably, so.
Yeah, that's also an option. But in general, it's like I can't take 6 weeks off, 7 weeks off, because it's only 6 weeks. What is that in the grand scale of things? What's that within the year? What is that going to be something detrimental to my 10-year plan, to my 5-year plan? No. Quite the opposite. It's going to help me to reset and not lose my love for the stuff I like to do. And again, coming from a sports background, I think many athletes, and especially swimmers, tend to lose the love for the sport because we're pushed so hard, so far, and at such a young age. Going back to training 8 hours a day. Yeah, we did it. We liked it. But at some point, it starts taking a toll on you. Not because you don't necessarily enjoy the training, but you train really, really hard, and then you don't see the payoff. And that's exactly what that like shitty month is. You try your best, it didn't work out. You see the end result, and you see the effort you put in. You put in 130%, and you only got out like 10%.
And it's hard to zoom out and see the bigger picture when your whole month is sucking. That can be a long time. All right, I think that's a good message for the end. Slowly we can wrap it up. I think it's been an hour and a half. I didn't even realize. Very nice talking to you guys. I think it's a very good message to sometimes, especially in academia, to zoom out a little bit and understand that having a bad month is, in the grand scheme of things, really just very short for the long-term goals.
It's not just a bad month. It's like a bad year, a bad 2 years. I mentioned at some point that it took me a while to finish my bachelor's. It took me 7 years to finish my bachelor's. 3 of those years, I did not get a visa. And that's like one of the big challenges when you're also studying abroad and you're like an international student. I couldn't change that. At that point, it was the most stressful period of my life. It was the most stressful period for my entire family. Like the entire family was like getting a single gray hair per second, like a new one just popped out of their head. Because everyone knew that I wanted to do this, and we ran into a wall that we couldn't break, we couldn't skip over, and we couldn't turn around from. Because at that point, it was like, I want to do this. And this is basically my only career. Because anything else is going to be a plan B, and I'm not big on plan Bs. So zooming out, looking at it from this point of view, yeah, it took me 7 years. Then it took me only 2 years to get to my master's. Who knows how my PhD is going to go? Who knows where life's going to take me? Hopefully, it's going to be like the average 75 years the average male gets. So I got time. I'm not going to stress in that case, especially not in science where your best years are going to be like your 40s and your 50s. And it's still going to be stressful, but at least you don't have to rush it to get to the most stressful parts. So I guess enjoy the little stress you have in your 20s or 30s because it's only going to get worse at some point. I'm not sure if that's necessarily a motivating message, but even if you have stress, you'll manage it. And in the grand scheme of things, it can always be worse. A lot worse.
I wish we touched on this topic a little bit earlier in the conversation about the non-traditional paths in academia, where, I think there's a lot of pressure on students thinking that, I have to, in order for everything to be perfect, I have to, from 18 to 22, it's a bachelor's, and then from 22 to 24, it's a master's, and then from 24 to 27 is a PhD, and you have to get it perfect. But if you, talk to professors who've gone really far in their research, like I've talked to several highly successful researchers, PIs in Europe and in the USA, many of them have very non-traditional paths. Some of them were moving around, their family was moving, or there was a war situation they had to escape. So their studies were obviously on hold. Or there was no money to finance their studies, so they had to work for a couple of years. In your case as well, you had a bureaucratic issue. You couldn't get a visa. So these things happen, and I think it's also a good message to see it again in the grand scheme of things as, not giving up on that path just because there's a delay in the moment. So I wish we talked about it a little bit earlier so we could elaborate on it, but perhaps for another time. Kajs, what is next for you? Where are you going? What's happening? You're finishing your exams. Is there a PhD in hold, or what's happening?
Well, one more exam, one more lab, and then we are officially done. And that'll be hopefully by the end of May. So roughly 3 more months. And as of right now, I'm preparing for a PhD, which will hopefully be in the Netherlands, more specifically in Groningen, where I'll be writing a project. That's the plan A, writing a project with my supervisor, getting the funding for the project, and then doing my own project, which would be the most amazing thing ever. And plan B is having a project from my supervisor because he currently has a grant which is awaiting approval, and if he gets it, the plan B would also be that I do the PhD as part of his project. So there's those two options, both tied to the Netherlands and both tied to Groningen. There's also some contingency plans, like I'll be throwing my application into the pot of the ETH PhD call just to see where things go, looking at some other options because I don't want to necessarily limit myself. Like, I'm big again on option A's, not huge on option B's. So I'm definitely putting all my time and effort into getting my own project and going down that route because it's the most rewarding for me, and again the most challenging. But I'm also wise enough at this point to consider other options, even though they might not 100% align with what I want. But I guess in life you got to consider the non-ideal situations as well.
All right. Good luck with all of that. So, as I said in the beginning, Kajs is writing a lot on LinkedIn, mostly sharing advice for students who are just a couple steps behind him. How can people find you? Is it just your LinkedIn, or do you have any other social media that you would like to share?
No, it's currently only LinkedIn because I consider it the main social media for the thing I'm doing right now and for the current step in my career. I did a lot of Instagram, I did a lot of YouTube. Some people might be aware of that as well. But currently it's basically just LinkedIn. And I think the one thing that I have to thank my parents for is the search term optimization of my name. I think if you just search my name, like my first name, you'll probably end up finding it within the first Google page or within one of the first results, which is kind of cool. But if you Google my full name, you'll definitely just find me and just no one else. So
So
thanks, mom and dad.
your future company will just be your name.
Yeah, basically, because why not? It's already optimized. I'm already there. I'm already indexed very well.
All right, perfect. Well, I wish you all the luck in the future with your PhD applications. Thank you for sharing your time, your tips, and your experience from your masters, from your bachelors. I hope this will be useful to people who listen to, and I hope this is definitely not the last time we talk.
Oh, definitely not. And again, thank you for having me. At some point we can revisit like those personal struggles and like struggles in general within another podcast and within some other topics to focus. But again, we kind of screwed away from our original plan as well. But yeah, that's just what happens when we talk science and get into it. But I think it's going to be useful for people in some way or another. And I think if they follow you on LinkedIn, they'll get a lot of useful info as well. So combine all of that, and they already got a lot of free resources.
All right, so just follow the two of us. You heard it, everybody. All right, everybody, that's the podcast for today. Thank you very much, guys. Talk to you some other time. All right.
I appreciate it.
Let me just stop this recording.